So hi, I'm Claudia Pahlke from the DKMS Life Science Lab, and I would like to thank Bhagya for the introduction. Topic of my talk is the ultra-high throughput HLA genotyping for stem cell donor recruitment. And in the next minutes, I would like to take you with me to the lab, and well, I hope you will enjoy it. So, which challenge are we facing? That's the impact of blood cancer. So every 35 seconds, someone is diagnosed with blood cancer worldwide. That means that more than 900,000 new patients are affected every year. For them, stem cell transplantation is often the only treatment option, but only 30% find a matching donor within their family. That means that 70% have to find another solution. That's why I would like to introduce to you the DKMS. It's an international non-profit organization. And it was founded in 1991 in Germany by Dr. Peter Haaf, who lost his wife Mechtel to leukemia. He promised her to help every blood cancer patient find a matching donor in the future. And so today, DKMS is the largest donor center worldwide, dedicated to the fight against blood cancer. So at present, there are more than 9.8 million registered DKMS donors. Then the DKMS was originally only active in Germany. You will find the most donors there. But now there is an increasing part of donors from other countries. So in red in that chart, you see the German donors. The yellow is US, green Poland, blue is UK, and very small is Chile in pink. And India is coming soon. So the DKMS enables 20 unrelated blood stem cell donations every day, so it's also coordinating them. That means that there are 11,000 second chances at life every year. So the DKMS donors in total have provided over 70,000 second chances at life to people in 54 different countries around the world. But nevertheless, 4 out of 10 blood cancer patients worldwide will not find an unrelated matching blood stem cell donor. So the story has to go on. Well, the question is, what happens when a person wants to register as a potential stem cell donor? All tissue markers which are relevant for matching have to be analyzed. And that's done in one single lab. And this lab, what I would like to present to you today, that's the DKMS Life Science Lab. So that's our ultra-high throughput lab in Dresden, so in Germany. So above you see our lab, and bottom our city, so it's worth a visit. And what we are mainly dealing with are human leukocyte antigens, or short HLA. And the system is responsible for the regulation of the immune system. So on all cell surfaces, the HLA proteins are expressed and they present foreign peptides to T cells or to the immune system. Genetically, the system is located on chromosome 6 in the HLA region, and it's functionally divided into class 1 and class 2 HLAs. So class 1, for instance, is H, A, B, and Z, and class 2 is HLA, DP, DQ, and DR. So the system is highly polymorphic. That means that there is an extremely low chance of two unrelated individuals to have identical HLA markers. And there are standard criteria which matches are relevant for transplantation, and these are HLA A, B, Z, DR, DQ, and DP. So here you can see now our milestones, so our type gene markers and our typing methods. So we started in 1991 with virology and analyzed HLA markers which were known to that time to be relevant. So and we changed to sequence-specific oligonucleotides in 1997 and joined the DKMS group in 2001. In 2004, we switched to Sanger sequencing, and the more genetic markers could be analyzed. But the biggest step forward was in 2013, where we change to next generation sequencing. Then many more markers could be implemented into our workflow. This progress is still going on. So there, for instance, is the blood group system, CCR5, KIR, which also plays a role. Some other HLA markers like DPA1, DQA1, DRP345, and also the cytomegalovirus infectious status, which also plays a role for the transplant outcome. So now I would like to show you our ultra-high throughput workflow. So let's start with the sample management. Though we have 24,000 incoming samples each week, and they are mostly coming as swabs, but there are also blood and DNA samples. And what we are basically doing is a sample prep for NGS analysis. So we have automatic data tracking, processing, and verification via our own database and our LIMS system. So new samples are integrated into our workflow. So one donor then corresponds to one well in the 96-well plate. At the first step, the DNA is isolated, and therefore we have 3 fully automated and integrated geometric magnetic separation modules, which operates unattendedly with an automatic scheduling of the plates. And here you can see our in-house integration with the geometric head on the left, the Beckman Coulter Bimec NX at the center for handling the plates, and the Lyconic carousel on the right for storing of the plates. And some more impressions from the biomagnetic separation you can see here. Next, the DNA is quantified using a TCAN platform for cytochrome measurement. And again, here we have unattended runs with an automatic scheduling of the plates. Then the samples are automatically sorted into high and low concentrated 384 well plates based on the DNA concentration. So that's called sample reformatting, and it's important for the normalization for sequencing as well as for the higher throughput. So one donor now is one well in the 384 well plate. Subsequently, the PCR can be prepared. So each sample is loaded onto different multiplex master mix plates. So in each plate there is one or more gene markers or multiple exons per gene per plate. And so one donor corresponds now to 14 wells in 14 different 384 well plates. So to sum up the pre-PCR workflow, you can see here our incoming sample types, so blood, buccal swabs, and DNA. They are portioned into plates, and then the DNA is isolated and quantified and reformatted and sorted into high and low concentrated plates. Then the PCR can be performed, and therefore we are using LGC hydrocyclers for an enhanced throughput, and they have 3 tanks with different temperatures according to the specific PCR protocol. And up to 32 plates can be moved from tank to tank by a robotic arm. After PCR, all genetic markers from one donor, which are located in 14 different PCR plates, can now be combined. And we are doing a second PCR to attach molecular identifiers, so-called MIDs, to the amplicons. So we have then a unique barcode on DNA level for each donor. And that's done on a Cyber instrument from Analytic Jena. Thanks to the MIDs, the samples can now be pooled. So all wells of a 384 well plate can be combined in one well of a 96 well plate. And they are then automatically purified by magnetic beads. And therefore we have 2 NX and 1 I5 instrument, which you can see here. So after sample pooling and cleanup, we are doing a quantification and quality check by QPCR. And therefore we have again an NX instrument. And the protocol is split into a dilution program and a preparation program to perform the QPCR in triplicates. After qPCR, the pools can now be combined, so in an automatic concentration-dependent way, to prepare them for sequencing library. So therefore we are using a BioMac-NX or BioMac-i5 system again, and now we have 40 pools, which corresponds to 8,000 donors, which are all combined in one single tube. And all of our programs are in-house developed and verified. And here we again have an NX or i5 instrument for doing best protocol. As the last step, we go to the sequencers. So we are doing paired-end sequencing using Illumina MiSeq, HiSeq, and NovaSeq sequencers. And we have our own demultiplexing and monitoring pipeline after sequencing. And most data can be analyzed automatically using our in-house developed genotyping software for high accuracy HLA typing called NextType. So to sum up the post-PCR workflow, we started with the PCR in 14 plates, and then all genetic markers of one donor were pooled together. And we have done the second PCR to attach the MIDs, and then we pooled many donors together to one pool, cleaned it up, doing a qPCR check, and then we combined different pools together for the sample library preparation and sequence them on a Illumina system, and then the data can be analyzed. So at last I would tell some words about the markers and why do we analyze more markers than the current state of the art. Well, the current criteria are recommend. So there's still significant room for improvement of transplant outcome. So the current state is HLA-A, BC, DRB1, DQP1, and DPP1. But many studies suggest that their donor selection can be optimized if you consider further HLA and non-HLA markers. And therefore, we fill the database with many other markers. As I already told you, there are HLA and non-HLA markers and cytomegalovirus status, which is pretty new, also possible from buccal swab samples. And so we fill the database to provide an upfront genotyping profile. And of course, further markers will become available easier and cheaper in the future due to ongoing advancements in sequencing technology. So we are also doing our own research. Here you can see a selection of our publications. So we also develop our own software, for example, the open source software TypeLoader 2 for an automated submission of new alleles. We are also doing research on new sequencing technologies like the Menai nanopore sequencer. We have a review about our workflow. Then we are analyzing so many samples. We find novel alleles which are unknown quite every day, and so we have a dual redundant sequencing strategy to confirm these new alleles. So if you're interested in more information, just visit our website, dkms-lab.de. So to conclude, we are doing next generation sequencing for more than 30 genes per donor. Therefore, we have 50 robotic platforms, analysis instruments, and sequencers. And we are doing around 24,000 new samples each week. That means 1.2 million per year. Or in other words, that means 65,000 individual PCRs on a regular workday. Therefore, we are the world's largest and most advanced HLA typing lab. And we are doing high-resolution sequence-based genotyping for stem cell donor registries. So with that, I would like to thank the whole lab for their contribution, and of course you for your attention, and I will be happy to answer your questions.